Okay, next generation sequence. We are Santiago Cuervo, Laura Guio, and Alejandra Garzón. So, introduction. The terms NGAs does not denote a single technique. Rather, it refers to a diverse collection of passenger sequencing technologies developed in the last decade. These innovations include sequencing by synthesis, sequencing by ligation, and sequences by ionic semiconductor and others. And in the image to the right, we can see that NGAs is a silent modified digital and vastly scaled up implementation of single sequencing. Mm, in both methodologies, a polymerase copies the input molecules by incorporating nucleotides from a pool that is either partially or entirely composed of tight and inextinguishable bases. Functioning rather than take advantage of size separating to organize fluorescent molecules, NGAs use positional separation. Millions of different temple DNA strands are attachment to discrete positions on glass slide and remain fixed in the same position through the direction of sequencing. Each template is then spread across a single modified base, and microscope captures an image that resolves both the position of each template on the glass, as well as its fluorescent color and intensity. The modified bases are converted to regular bases so that they become extendable and non-fluorescent.
Okay, features. NGS-based tests have revolutionized the study of genome by allowing the reading of millions of DNA sequence in a massive parallel manner in a shorter time and the lower cost per base. These tests include gene panel sequencing, whole exome sequencing, and whole genome sequencing analysis of their result in complex and required thorough bioinformatic and clinical process for proper interpretation. Limitation of NGS tests includes technical aspects such as coverage, depth, and length of sequence, which can be avoided by implementing good laboratory practice. NGS sequencing bias tests are diagnostic tools that must the based on adequate clinical approach for their reason use correct interpretation and correct decision making. It is a great importance that doctors have the basic information to be able to request tests given their current clinical relevance.
This technique have a lot of application. For example, genomic, economic. Um, the NGS technology make easier and more possibility to identify genetics and responsibility for additions. It revolutionized the way both biological and evolutionary process are studied on molecular level. Um, for another hand, have a study on the food safety because facilitate identification of genomic variations in areas of concern and allergens, bioactivity, some the toxicity of anyway expression products like fruits and vegetables and other food. The another application is in transgenic technology. In transgenic means use the study cellular and tissue specific response to the size by modifying the DNA sequencing or transferring the DNA to the animal on certain proteins can develop and one use the medical treatment. Um, the most popular application is human health because use this technique in a study on cancer and similar and clinical health and some studies for use examining someone microorganisms in the body. And these are the bibliographic.